we lysed HeLa cells and we took from them all of their proteins, and we called that a HeLa whole cell lysate. And now we want to know how much protein we got back. So what we're going to do is we're going to run a protein quantification, starting by making a standard curve. 6 tubes labeled 0.4, 0.8, 1.2, 1.6, 2.0. And we're going to pipette into each one of these tubes a certain amount of something called BSA and another amount of water. So if you're following along in your manual, these are the amounts that we're going to be pipetting in. So we're going to start with 0, bovine serum albumin. Bovine serum albumin is a known protein, and we have 2 micrograms of protein per microliter of sample. I'm going to pipette the rest of these and then come back to you, but what I'm going to show you is that the very last one is going to have all bovine serum albumin, all BSA. So I will have 2 micrograms per microliter of protein in this tube, and I'm going to create a dilution scheme. So I pipetted BSA so that it got higher in concentration until I reached 2 micrograms per microliter. I'm done with my BSA, and what I'm going to do is I am going to vortex and bring into solution all of these standard. So I'm going to set this standard up with my protein quantification in a 96-well plate. It has 12 columns at the front, the top. It's actually numbered 1 through 12, and then it has A through H along the side. We're going to follow a scheme, and I'm just going to simply go about the business of pipetting in 50 microliters of each one of these standards according to my preset scheme. I actually take my 50 microliters and I drizzle it into the side of this well rather than pipetting to the bottom, because I'm afraid I'm going to squirt it up, and I'll just do that 3 times over for all of them. So I've reached my last one, and you see how I move back the ones that I've completed, and I'm going to grab this and then into my row, my column 6, A, B, and C, I'm going to install 50 microliters, and I'll be done setting up my standard curve. And now before I add Biuret to anything, I'm going to set up my sample. I'm going to, into column 9, add 50 microliters each of HeLa. But before I do that, I'm going to create a 1 to 5 dilution, because I'm pretty certain I'm going to have way too much HeLa whole cell lysate. So I'm going to add 160 microliters of water and 40 microliters of sample. So I added my 40 microliters of sample, and I'm going to dilute it with 160 microliters of water. So now I have 2 samples that I'm going to load up into my dish, into my columns 9 and 10. So I'm going to put 50 microliters of HeLa neat into